Hello, my name is Mary Regina Boland. I'm an assistant professor of informatics, and I'm going to tell you a little bit about my research on methods and considerations for studying neighborhood effects on human health using electronic health record data. For disclosures, I have no relevant relationships with commercial interests to disclose, and I am a member of the Genomics and Computational Biology, or GCB graduate group. I am also a member of their Committee of Equity, Inclusion, and Diversity group. I am also a member of the graduate group in epidemiology and biostatistics, and I am also a BGS Prevail Ambassador Committee member for outreach and engagement. Please feel free to reach out if you have any questions or if you're interested in rotation projects in my lab. So a little bit about my work. I do a lot of work with electronic health records, and electronic health records include information collected during routine clinical care. Everything pretty much you could imagine ends up being recorded in electronic health records, assuming that you end up in the hospital. So there are actually codes for whether or not you're injured or hurt at the library, walking into a lamppost, or being struck by a sea lion. And we are able to use this information to study both disease prevalence and incidence rates, as also to look at where people live and neighborhood level effects. Do a lot of research on environmental exposures of this nature. And we have two papers shown here, one on the left on COVID-19 and one on the right on severe maternal morbidity. A big feature of the work in my lab is on maternal health outcomes. That's a big part of some of the work that we do. A previous PhD student in actually GGEB, she was in epidemiology. She graduated this past May, named Jessica Meeker, and she developed a method for using electronic health record address data to identify residential mobility, or whether or not you moved from one location to another, using electronic health records. And this was actually published in the International Journal of Epidemiology, which is the top epidemiology journal. Firstly, the importance of neighborhoods. This is a beautiful picture of Philadelphia in its finest. There are some very rich communities just outside of the city of Philadelphia in Gladwin, which was the sixth richest zip code in the entire country, the US, if you base it on real estate values. And then also Chestnut Hill, Philly is one of the richest areas within the city of Philadelphia, with an average home price of $750,000. Likewise, there are communities within the city of Philadelphia that are very poor and impoverished communities. Here's some pictures of those communities as reported by WHIY and other news media. And you can see visibly that social distancing is not very easy in poor communities. Also, there's a lot of environmental exposures in terms of paint and dilapidated structures. There looks like there's some evidence of individuals living in the middle home over here, up in the top, but obviously it's not really a habitable environment. So we're able to look at spatial association analyses to understand neighborhood level effects and their effects on SARS-CoV-2 positivity, which is basically a positive COVID-19 test, but you might not be displaying symptoms of the disease. So our method was to use the American Community Survey and Open Data Philly to identify neighborhood level factors at the census tract level. From Penn Medicine, we were able to get positive and versus tested patients with symptomatic status, perform some spatial regression at the neighborhood level to understand the effect of SARS-CoV-2 positivity and asymptomatic only. This would be useful for further resurgence planning in the future. Our overall SARS-CoV-2 positivity analysis, what we first did was tests for significance between each neighborhood level characteristic and SARS-CoV-2. And this found a lot of significant neighborhood level features. We then put those features into a univariable model that wasn't univariable anymore. It was multivariable. So we took all of the positive ones at the univariable level and put them into a larger model. And in that larger model, everything is adjusted for all the other significant factors. And so we were able to find 4 neighborhood level factors that were statistically significant in SARS-CoV-2 positivity rates. 2 neighborhood factors increased the risk of being positive and 2 decreased the risk. So the things which increased risk were having at least a high school education, which is our proxy for education status, and identifying as Hispanic or Latinx in a community. Factors which decreased your risk were proportion of individuals at a census tract level identifying as white alone, or individuals receiving public assistance. So interestingly, living in a neighborhood receiving public assistance tended to lower the odds of SARS-CoV-2 positivity in the fully adjusted model. So what does this mean? So public assistance includes cash and non-cash benefits, so TANF and SNAP, to low-income families or individuals. And we believe this could be associated because these programs provide food and financial assistance to needy individuals and families, and this in turn can reduce their need to work and go out during the pandemic. This enables social distancing efforts to be more effective. We're not 100% sure if that's the mechanism, but that is our proposed explanation as to why this was protective. Our overall conclusions from this work was that neighborhoods do matter, public assistance is important. We do not believe race and ethnicity was associated because it is a biological variable. We don't believe it was a biological variable in this study, but is rather a proxy for social class and could be due to riskier jobs based on social class, housing conditions not otherwise captured in our models, and various other factors. And it is important just to note that, again, we're not considering the race associations to be biological in nature, but again, to be proxies of these other conditions of the human experience. Considerations with regards to electronic health records. So Jessica Meeker, our now Dr. Meeker, developed this method for classifying whether or not individuals moved, and it's called REMAP, and it's published online, and it's really useful because it's an algorithm that can identify whether different electronic health record addresses are in fact the same. I'm not going to go into the nitty-pitty details of the algorithm, but we're able to have different decisions to determine whether or not the town was actually the same or the location was the same and so forth. She manually reviewed over 3,000 addresses, and we found an accuracy of above 95%. Interestingly, we did find that 41% of pregnant patients moved during their pregnancy within the city of Philadelphia. This was more similar to certain surveys that have been done, but lower than other estimates in other work. And we were able to outperform using zip codes alone because a lot of times individuals moved within a particular zip code. The effects that this can have on misclassification are huge. People do not move at the same rates within the city of Philadelphia. And also people move, when you look at neighborhood deprivation as a score from 0 to 100 and also 0 to negative 100, people don't always move to better neighborhoods. They also move to poorer neighborhoods in some cases. So what we were able to find is that there was change in deprivation score for about 1/4 of pregnancies. Some moved to higher or poorer neighborhoods, some moved to less poor or less deprived neighborhoods. About 11% moved to a more deprived neighborhood and 12% to less deprived or a wealthier neighborhood. It is important for AI and machine learning because when setting a threshold, you need to understand that if you didn't take into account moving, 24% of our patients would have been misclassified. And this can be very important when you're studying environmental exposures during pregnancy. And this code is actually available, and feel free to email us if you're interested. She also did some work on the effect of neighborhood deprivation on post-induction cesarean section, showing that the more deprived neighborhoods had the highest rates of post-induction C-section. This is really important work from women's health perspective. And those are the adjusted odd ratios. Again, if you're interested, please send an email to me. I'll be happy to tell you more about my work and more about how it might fit in with some of the different graduate groups that you may be actually more involved in. And I just want to briefly say, here's a couple pictures of my lab before COVID when we were actually able to socialize. This is now 2019, almost 2 years ago. Hopefully, things will end soon and we'll be able to meet again more in person. This is my lab. I present a lot of work from Dr. Meeker, who's now at the CDC. She graduated this past May. A couple of the individuals in these pages have graduated, but some are still in the lab, particularly Dr. Sylvia Canalong and soon to be Dr. Aditya Kashyap are still in the lab, and some of the undergrads, as well as Lena Davidson, our data analyst. If you have any questions, please feel free to reach out, and I look forward to hearing from you. All right, thank you. Bye.